No, ma'am.
Okay. So today we are going to discuss about the database. Like our second module, we are going to start from today. That's bioinformatics databases. In that, the first we are going to discuss about the NCBI and the GenBank, means like a gene database. So first and foremost, I will just give you the overview about the NCBI. If like from you, if somebody is new to the NCBI, it will be helpful for you. So NCBI, that stands for National Center of Biotechnology and Information, which is developed or and maintained by the National Institute of Health and the National Library of Medicines, right? So NCBI actually it's not a database, it's a resource you can say where the collection of the databases are present. Means whenever you are searching in a NCBI, so what kind of data you want to search, accordingly you need to be select the database over here. Means let us consider if you want a gene information, you can go with the gene database. If you want genome information, there is a genome database. If you want articles, there is a PubMed database is there. Then, if you want a protein information, protein, raw data for the, then here we are having the SRA, then taxonomy for the taxonomical data, structural. So every database, like most of the database here, you can are present. So what kind of data you want to retrieve, accordingly, you have to select the databases. So once you have selected the database, after that, here you are having the search box. Now, whenever we are searching in a database, I'm talking generally, whenever we are searching in a database, there are two types of searches we are having. One is a text-based search, another is a sequence-based search. So text-based search means what? Like where you are having an ID, where you're having a name of your entry, you're having the accession ID, you're having a location, or you're having some keywords for it. So that's what we call it as a text-based search. And another we are having a sequence-based search where we are giving a sequence as the input, right? So this NCBI is a text-based search. It is using a text-based search method to search a data inside it. And here you can give the name, you can give the ID if you know, you can give a keywords, whatever you wanted to type, organism name. So this is how basically you can search in the NCBI here. Now, in the right side here, it is giving the popular resource. Like if one of the resource, if you wanted to access quickly from here, you can go with it as they have used maximum times or they for this resources, people are searching maximum time. So that is what here they have given as a popular resources. And in the left side, the total home resources we are having like chemical, bioassay. Then here you're having the database, sorry, data and softwares. Then you want a data regarding the DNA, RNA, or genetics and medicines. You want a literature or protein or sequence analysis data. So this home resources is given over here. Then in the right side, the NCBI news and blog section is added. So whatever the new news you are getting, then any blog if you are getting that, you will be able to read here. And in the middle side, we are having these six different tabs. These are nothing but the six different functions of NCBI. As you can say, in NCBI, NCBI is an open source resource where anyone can submit the data. So maybe if you wanted to submit your data or your manuscript inside the NCBI database, you can go here with the submit option. You can just select uh, the query that what you want the sequence or whatever the type of data you are having. You can just select that, you can follow the procedure, then you can fill the form and then you can easily submit the data inside the NCBI database. Then the next function we are having here is a download. As majority of the NCBI data is available here for the download and that is completely free. you can easily transfer the data inside the NCBI into your system. So what kind of data? It can be sequence data, it can be tools information, it can be any tutorials, anything. You can, I'm not saying all the data, but most of the data is freely available. Some data, if they are in a controlled access, that you cannot download. Okay, so or maybe if you wanted to download a complete NCBI data using the FTP link, because every database is having a FTP link where all the data is stored inside the FTP link. So if you wanted to download that complete information, you can go with the NCBI FTP site also. So this is how you can download the data from NCBI. This is what a second function. Third is a learn. Actually, I don't know you have heard it or not, but NCBI is arranging some seminars and webinars, right? So if you wanted to attend their seminars, also they are creating a variety of educational products like courses, workshops, webinars, training materials, their documentation, and it's completely free and open to everyone. They are having a free educational events. So if you wanted to use that educational materials, you wanted to download that courses, their workshop, like you wanted to attend their workshops, or you wanted to get their training materials, tutorials, so you can easily get it from the NCBI and you can reuse it also, you can distribute it to others. So this is what a learn section. Then fourth you are having is a develop section. Actually, NCBI is not only like a database, it's developed a variety kind of tools also. So maybe if you wanted to develop your own tool and you wanted to access their APIs, okay, you wanted to access their API, that is application programming interface, or you wanted to use their code libraries to build your own application, so that time you can go with this develop section. You can see that it is providing a variety of resources that allow developers to access and manipulate the NCBI data in their application. Means let us consider you wanted to build some databases also. You need some information from NCBI. That also you can take if you wanted to develop a tool, and whatever the tools it is having, if you wanted to use some APIs from that, some code from that, you can take it from this develop section. Then analyze section, as NCBI having a variety kind of data analysis tools which is used for handling the data, then manipulate the data, visualize the data. So that you can check it from the analyze section. You can see it is providing a variety kind of data analysis tool for manipulate, align, visualize, and evaluation. Also, the tools is also given here, the names, all the tools. If you want and the section also given here, if you want specifically literature related tools, if you want health related, genome related, then genes related, protein and chemical. So this is how a different section also given. So which tool you want, from which category you want, you can check it with that particular category. So this is what a analyze section. And the last is a research section. Actually, NCBI, they are having a collaborative projects. So maybe if you have any research idea, you can send them your idea, and if they find any interest, they can contact with you and you can collaborate with them. Right? They are having such kind of research programs, which is carried out by senior investigators, then staff scientists, then fellow students. So if you wanted to collaborate with them, here it is given their information, the address, phone number, everything. Then tools and topics also you can select it from here. Then they are giving their information of researcher groups, staff, seminar schedule. So everything is given over here. You can check it out, right? So this is what basically the 6 different functions of NCBI. And this is just a normal overview of homepage, because if you are new here, you should know all these things, right? So let's start with our first topic now. That is GenBank database, or you can say a gene database. Actually, a gene database is a public database of nucleotide sequence. which is supporting to the bibliographical and the biological annotations. Means you are not getting only a sequence, you are getting the bibliography also, means you will get the articles and all related with it. Along with that, you are getting the biological annotation. Biological annotation means what? You will get a complete information about it, like in which function it is involved, to which protein it is coding, to which protein it is interacting, in which pathway it is involved, what is so everything related with the gene, you will get it in the gene bank database. It was built and developed by the NCBI. It was created in 1979 at the Los Alamos National Laboratory. So at that time, it was called as a Los Alamos Sequence Database. But later in 1982, it was renamed as a GenBank, and that time it became a public database. Okay, so this is just the introduction. Now, whenever we are talking about the GenBank, actually, I have explained you the primary and secondary databases. So, primary and secondary databases, here we are having a three databases of nucleotides: GenBank, EMBL, and DDBJ. So, this GenBank is also one of the primary database like EMBL and DDBJ. And what content it includes? So, it contained the genomic information like DNA, RNA, cDNA, raw sequence data, and many more things. Just what I have told you before. It contains a complete information about the gene, to which protein it is coding, to which RNA, like what RNAs are expressing, everything you are getting here. How many RNA it is forming, so that complete information you will get in the NCBI, so GenBank database. So it is using a text-based search method. And whenever we are talking about the GenBank file, means like a GenBank data, so it is giving you a one file with one complete information inside of one file, and that file you can download with the .gb extension. Just like I have told you earlier that if you wanted to download a complete sequence from a database, it is in a faster format, right? So whenever you are going to download a GenBank file, it is having a .gb extension. But what information you're having in the GenBank file? So basically, the GenBank file is divided into a three section: header, feature, and sequence. I will explain you this now, but before that, do you have any question? You wanted to ask something?
Hello. Ma'am, please explain primary and secondary databases.
I have explained it earlier, so you didn't attend that session?
When, ma'am?
In the very first session. Can anybody explain the difference between primary and secondary database?
Ma'am, primary database is the database which is open source, whereas secondary database is the database which has data which is like correlated and corrected by the agency itself.
Yeah, like they are having the expert panel and they are giving us a cross verified data, which is also called as a curated data. Okay. So it is a request to all of you that whatever the recordings we are providing, at least a one time please go through the recording, so that whatever the concepts we are like taking here, it will be more clear to you. I can understand in one go you will not able to recall all the things. So at least 1 hour you can give to revise all these things by rewatching the recording, right? So please go through the recordings. Okay. Uh, is it clear now?
Yes.
Next thing is like let's talking about the uh, GenBank database file, which is divided into a three section: header, feature, and sequence. So the header, it means a summary section. You can also this as a summary section where you will get a summary of your entry, like what is the locus, what is the description, then what type of entry it is, whether it is DNA or RNA, then what is the length of it, then what is the accession, version, organism, reference, author, title. So such kind of a summary information you will get here in the header section. Is it visible? Can you see this header section here?
Yes, ma'am.
Okay. Then next you are having is a feature section. So the feature section is what it is containing the actual data which we required. That's actual feature of your entry. Means maybe if it is a gene, so in the gene, what is the start and end position of the gene on the human genome? Then how many RNAs it contains? So if it's contain the RNA, what is the position of that RNA? Then what is the product of the CDS? What is the length of the CDS? What is the product of the CDS? So that every feature information you will get in the feature section. This section is very important for us, feature section, because here only you will get a complete information. And the last section you are having is the origin section, where you will get a sequence. But this origin section is not the FASTA sequence. It's just the sequence you are getting, because FASTA sequence is always started with the greater than symbol. So please do not confuse between the FASTA sequence and the sequence in the origin section. Okay. So this is how
Ma'am,
basically,
can you explain the feature section once again?
yeah, feature section basically we are getting the actual features. Means let us consider you are having a gene. Right now you are searching a gene. So in that gene, uh, how many exons you are having? Okay, so let us consider you are having a 5 exons. Okay, so all that 5 exons you will get in the feature section along with their start and end position in exons and introns both, along with like the name. Maybe if it is a coding, so what it is coding, so that complete information you are getting. Just like here, the example they have given, this is a CDS region they have given, right? So the CDS region, it is from which gene, what is the codon, product is what, protein ID is what. So everything you will get here in the feature section, the actual features, what it contains. Okay?
Yes, ma'am.
Yeah. All right. So I will show you practically whenever we are performing the questions, this GenBank file. But as you should know how it is look like, how it is looking like. So that's what this example we are taking over here. So this is just a theoretical part, but whenever we are going to perform it practically, it will be more clear to us, right? So now we are going with the questions, the exercise question. You all have to perform along with me. Okay, means I will perform first, I will give you a time, and then you can perform. All right. And I will give you some task also, so you can try that questions uh, after the session. So here we are having search for the gene PRNP for Homo sapiens. Like first thing, we need to search the gene. Now how we are going to search the gene here? I'm going to type the name PRNP. Okay, that's what I wanted to search. But if I will click on the search, is it giving it? It will give me a better, like a proper result. It will give me a entry of PRNP gene.
Ma'am, you need to select the database. What kind of?
Correct. Yes. We need to select the database, right? Because the question is we need to search the PRNP gene. So first you need to select the database, then you have to type your name, your entry, what you want, and search it. Yeah. So whenever you are searching for the PRNP here, on the in the box you are getting one entry PRNP, that is a prion protein, that is from the Carno blood group. Then this is a full name. Here it is giving the name of organism Homo sapien, because as per the question, we want to search the gene for Homo sapien. Is the question is visible?
Yes, ma'am.
We want to search the gene for Homo sapien. Now, if you will see, uh, this is also given about the Homo sapien. In the right side also, they have given the different organism. So maybe whenever you are searching for the PRNP, you want from some another organism, the list is given over here. You can check it out from here also. Otherwise, if you will just go down, here you are having the table where it is giving the PRNP gene for different organism. Like this is the PRNP for Homo sapien, this is from Mus musculus, this is from Rattus, and many another organisms. So you can select from here. So the choice is completely up to you, that you can open it from here also, or from here also. Okay. So I'm just going to be click here, and it is
What
going
is
to be
1145 there?
1145 actually, because this NCBI is an open source resource. So for a single entry, maybe there are multiple submitters, right? So these are the multiple entries. But out of that, not all the entries are validated, right? Maybe they can be not, they are not reviewed, means they are not cross verified, right?
Okay.
So you need to check, maybe from this 1145, you're opening off, you let's consider you're opening some entry which is at 100 number. But you have to see that whether that entry is correct or not, means whether it is cross verified or not.
So where can we see that?
Yeah, on the page here itself, whenever we are going to open it.
Okay.
So this is opening the complete report, a full report of the PRNP gene. So here it is giving a name and the full name, like a prion protein, Carno blood group, Homo sapien, human. Then here it is giving the gene ID, because every entry in the database is having a specific ID. So the gene ID of PRNP is this. Then updated on. So actually, NCBI is maintaining the pages or updating the pages day by day. So whatever the last updated date, that is given over here. So it's updated lastly on 19th June. Okay. Then in the right side, they are given the table of contents. So this is just like an index. Means let us consider you want a variation. So you just have to click on the variation and it will navigate you on that section. You don't need to scroll it one by one and search where is the variation. So this is helpful to search the different sections. Then here in the summary, so the first section, if you will see, that is a summary which is giving you the official symbol, that is PRNP, which is provided by HGNC. HGNC, that stands for Hugo Gene Nomenclature Committee. So there is a one committee we are having which is giving a nomenclature, official names to the particular entries in the databases. So this nomenclature committee is the Hugo Gene Nomenclature Committee. If you will see, like keep your cursor here, it will give you the name. Then official full name, that is also provided by HGNC. Then primary source, like from which source they have taken the entry. Then see related information. So the related information you're getting from the another database also, if you want, like Ensemble, MIM, then Alliance Genome. So if you want to see the information of PRNP from another database, the link is also provided here. Okay. So this is how basically some information we are having. The gene is a protein coding gene. So this is some summary section we are having here in the first section, information about it. Then next question we are having.
Ma'am, You were going to tell how to check if the entry is validated.
Yeah. This is given in the question. So yeah, we'll come up to it. Okay. Uh, or maybe as of now you are asking it, you can see the RepSec status is reviewed. It is given directly. If it's not reviewed, it is given unreviewed. Or criteria is provided by single submitter, or criteria is provided by multiple submitters. So this is how you will get. Okay.
Yes, ma'am.
Yeah. So let's go with the next question. On which chromosome it is located? So here, if you will go down, actually everything is given inside the question. So we are going to discuss each and everything here. So now let's talking about the chromosome location. Here, it is given the location. Can you elaborate? Can somebody please elaborate what it is mentioned? Like 20p13, what it is?
Ma'am, it's the 20th chromosome P arm. That is
Mm-hmm.
the longer arm.
Mm-hmm.
And then the 13 is for, I guess it's 1-3 or something like the sub-regions on the longer arm.
Okay. Actually, P is not a long arm.
Yeah, ma'am, it's shorter. I got
Yeah. Okay. Anyone else? Sonali ma'am, Tanya ma'am, Annapurna ma'am. Okay. So to understand this, we'll see one image so that it will be more clear to you. So if I will show you this image, is it visible? Can you see this image properly?
Yes, ma'am.
Yes, ma'am.
No, 1 second, sorry. Yeah, I think now it's properly visible. So here they have taken the example of a CFTR gene, right, which is a same, and it is giving a same format what we have uh, discussed in the PRNP page. So 7, as so you correctly said, it's a chromosome number, as we have 22 chromosomes and one XY chromosome. So this is located on 7th chromosome format. Then Q arm. So there are two arms of chromosomes we are having, a short arm, long arm. Short arm is a P, long arm is a Q. So it is located here on the long arm, Q arm. Now 3 is a region on the long arm. 1 is a band on the region, and after the dot, whatever you will get, that is your subband. Okay. So now, can you tell me what it is representing? Instead of Arya sir, is there anyone who can tell me?
It is the
So
20th
the
chromosome.
Yeah.
So the gene is on 20th chromosome, short arm, and
Correct.
and it's
Correct.
the first region. It is on the first region of the short arm
Correct.
and third band of the first region.
Good. Yes. So it is located on 20th chromosome. Three arm, short arm, one region, three is a band. And maybe after the dot, if you have some number, that is what your subband. Okay. So this is how we can understand that it is located where on the chromosome exactly. Is it clear to everyone? Because this is going to be uh, come again in some another database. So at that time, you should able to know, you should able to tell me what it representing. So if you have any question, you can ask me right now only. Okay. So let's go with the next question. What other names this gene is having? So other names means nothing but the alternative names. And here, if you will see, it is showing also known as. So in the also known as, here it is giving some alternative names of the gene, because previously for a single gene, we are having a multiple names according to the different regions, different countries. But right now we all are having the official name, which is PRNP. And for each and every entry, there is an official name we are having right now. But maybe in some older research papers, you might get these names. So this is nothing but the PRNP only. So at that time, maybe if you are getting the gene name Puru, so you need to be check what is the official symbol of that. Okay, is it clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Uh, then how the gene is originated. So it is asking the origin. So can you tell me by which section we can get the origin of the PRNP? How can we get the origin? How it is originated?
From the lineage.
Right. So from the lineage is nothing but it is showing you how the organism is originated, how it is falling under the different taxas, that we can get it by the lineage. So how it is evolved from eukaryota to the Homo sapien. So that complete lineage it is given. Okay. So this is how basically the PRNP gene is originated. Is it clear to everyone?
Yes, ma'am.
Yeah. Any doubt to anyone else?
No, ma'am.
Oh, what is the function of the gene? So how can you get the function? You can see here in the summary section. In the summary, the function of the gene is given that it is associated with the Jacob disease, fatal familial insomnia, Huntington's disease, Kuru, and many things. Then here the summary information also it is given to, it is found on which chromosome, what is the size we are having for the gene on upstream. Then it is biochemically and structurally similar to the protein that encodes by the gene. Then what is the mutation status? Mutation in the repeat region as well as elsewhere in this gene have been associated with the Jacob disease and like these diseases. Then here you can see the overlapping open reading frame has been found on the gene that encodes a smaller structurally unrelated protein, ALT-PRP. So here in the summary section, you will get a complete information. And also here you will get it is submitted by the RefSec. Okay, so if you want theoretical information or functional information, you can just read out the summary. All right. Then what is the RefSec status of the entry? So the RefSec status here you can see it is a reviewed. RefSec actually is what? It is one of the database which is providing a status to the entries. So if the RefSec status is reviewed, it means you can directly use this information for your research purpose as it is cross-verified as it is validated information. Okay? If it is not reviewed, you will get as unreviewed, or if it is like a provided by single submitter, provided by multiple submitters. But if it is validated, you will get a reviewed. So this information you can go directly, you can take directly for your experiment purpose or research purpose. No need to cross verify it, no need to validate it. Is it clear?
Yes, ma'am.
Any doubt? Anything you want to repeat? You want to repeat up to now? Any question you want to repeat up to now? Till here? I'm asking to everyone. It is not possible for me to take the names of each and everyone. So please tell me.
Ma'am, I have a doubt. Can you explain me that chromosome thing again? I couldn't understand that well.
Yeah, sure. So here, this is what you're saying, right?
Yes, ma'am. I'm actually not aware how to read this chromosome thing, the short arm, long arm. Like,
Okay, okay.
yeah.
So if you're talking about the chromosome, so the gene, so here it is giving the format. So 7 means what? This CFTR gene is located on which chromosome out of 22 chromosomes and X and Y. Okay? So it is located on 7th chromosome. Now, whenever we are seeing the chromosome structures, it is divided into two arms. So there is a one short arm we are having and there is a one long arm we are having. So short arm that given a name as a P and long arm is a Q. So it means this CFTR gene is located on long arm, that is on a Q arm. Okay? Because on a chromosomes we are having a multiple genes. So it is located on Q arm, not P or not a short arm. Then third, so this 3 is a region in the long arm. There are different regions we are having. So it is located on third region. So in the third region, it is located on first band. Okay, on the chromosomes we are having the bands, right? So it is located on first band, and in the band it is located on second band, sub band, sorry. So after the dot, whatever the number you will get, that is your sub band. Is it clear now?
Yes, ma'am. Got it.
Okay. Maybe you can just take a screenshot of this image by searching the chromosomal location. So it will be more clear to you whenever you wanted to read such kind of locations. Okay. Anyone else, like any another question you want to repeat? Okay. Now, next question. What is a strand information of gene? What is a strand information means? On which strand it is located, forward or reverse. Now, here if you will see, it is given the chromosome 20 image. Let me just zoom the image here. It is giving the chromosome 20th image, and here the PRNP is highlighted with the dark blue color. Along with that PRNP, we are having some another genes. These are the nearer genes of PRNP. These are what? These are the nearer genes of PRNP. It's not like that on a chromosome 20 we are only having a this much gene, but it is taking a section from this base position to this base position only. And from this, whatever the genes they are, it is coming, they just highlighted over here. Now, can you tell me what is the direction of PRNP? forward or reverse?
Is it forward?
It's forward, right? Because you can see the direction, the arrow, it's from the 5 prime to 3 prime, right? It means it is located on forward strand of chromosome 20, correct? Are you getting my point?
Yes,
Yes,
ma'am.
ma'am.
Now, if you remember, I have told you one thing about the NCBI database, that NCBI database is always storing the information in forward manner, right? But here you can see that it is a forward direction, that's fine. It is giving the direction I want a base position. Means on 20th chromosome, from which base to which base the PRNP gene is located. Okay. So to get that exact location, here you can check this table which is given. So this table, you can see it is showing the annotation released, means the information which is released, that ID it is given. Then here it is giving the status, that is a previous assembly and current assembly. So assembly is what that we have discussed, which is a collection of the genomic information. And whenever the day by days assemblies are updated, the information is updated, assemblies are also updated. So here they are comparing between the previous assembly and the current assembly. It's not like that in human we are only having a two assembly, but for now we are just comparing between the previous and the current one. This is the current, because there are two current assemblies are given. You have to focus on this first and then previous. So the previous assembly, it's GRCH37 assembly. And here, this is GRCH38 assembly. So this is the ID. Previous is a 37, current is a 38. And whenever we are retrieving the information, always we have to retrieve it from current assembly. Okay, 38. Now here it is giving the chromosomes, like it is located on 20th chromosome. Now here it is giving the NCID. NCID, that stands for NCBI chromosome. See, every database, whenever you are searching a ID in any database, every database is having its own prefix. Just like for NCBI chromosome, the prefix is NC. Just like NCBI mRNA, there is a NM. NCBI protein, there is a NP. NCBI non-coding RNA, there is a NR. So this is how basically we are having a prefix. So you can understand whether it is a chromosome, whether it is a mRNA, whether it is a non-coding RNA or it's a protein. So by that prefix you can understand. Then here you can see the coordinate it is given. Coordinate, that means a base position. So on 20th chromosome, PRNP gene is located from this base to this base, right? So this is a location, base position, you can say, of PRNP gene on 20th chromosome. And if you will see, if you will compare it between the previous assembly and the current assembly, you can see the change in the coordinates, right? But always take this from current. This chart is clear.
What is the second line showing?
Second line is a T2T assembly. That is a telomerase to telomerase assembly.
Okay.
Yeah, this is not a whole genome assembly.
Okay.
It just released few months back. Previously, it was not there. Yeah. Any another question? Because every time they have not provided you the diagram, right? Like it's a forward strand, like arrow wise, they are not providing you. They will give you the base positions directly. Correct? This is clear because I want to explain some more about it. But before that, this should be clear.
Ma'am, NC is the code for the
NCBI,
database.
NC is a prefix of chromosome in the NCBI database.
Prefix for the chromosome.
Right. If you have
And the
mRNA,
so that is the okay, you were telling about mRNA.
yeah. So if you have mRNA, you will get NM as a prefix and the ID in front of it. If you have
Okay,
protein, you
and
will
the
get NP.
decimal 11.
Decimal 11, these are the version number. Like this is the previous version, this is the latest version.
Okay.
Okay.
But in both these assemblies,
Hmm.
chromosome number is changed 37, 38.
No,
Then the
no, this
region
is the
was
assembly ID. This is not a chromosome number.
okay. So
This
because
it,
P13, P14, they are nothing to be
no,
confused with.
these are the patch 13 and patch 14. I
Okay.
will explain this patch concept in UCSC very detailedly. But for now, you understood that the chromosome column is given here. You can see CHR,
Yeah.
that's chromosome 20 only.
Okay.
Chromosome is same, just the coordinates are changed.
Okay.
Like there is a slight changes. Obviously, you are getting from the assemblies to assemblies, not like a complete chromosome will change, right?
Right.
Okay. Now, my question is here, PRNP gene is located, luckily, like right now, whatever the gene we are getting for study, we are checking here for study is luckily located on forward strand. But let us consider there is a one gene you are having. Consider the gene which is given here PRNT, or the gene, there is some gene, a random gene, which is located on reverse strand. Arrow wise, you will get the information, that's fine. But maybe if they didn't mention the arrow, they have just directly mentioned you the coordinates, like on the 20th chromosome, the PRNT gene, which is given here, if you will see, it's a reverse strand, right? So it is located from this base to this base, the base position they have given. But one thing which I have told you earlier in the first session, NCBI always storing the information in forward manner. So the coordinates also you will get as per the forward manner in the increasing order, right? Because if you remember, I have taken one exon example, right? At that time, the number of exons for the reverse strand is also in an increasing order, and for the forward strand also in an increasing order. But ideally, it should be in a decreasing order for the reverse strand. But in NCBI, we don't have that case. So my question is, in that case, if the coordinates is given only, how can you understand the gene is located on forward strand or on the reverse strand? Because for both the strand, coordinates will be the same. So now,
Ma'am,
how,
ma'am, the
first of
second
all,
coordinate
my
will be
question is clear. First of all, tell me, is my question is clear to all of you?
yes, ma'am.
Because yeah, if the question is clear, then and then only you will
Yes,
be able
ma'am.
to understand. Okay.
Yes, ma'am.
Yeah. Now tell me, any guess?
Ma'am, the second coordinate will be smaller than the first one. Here,
Right.
ma'am, like it will be less than the first coordinate. So we'll understand that it is not in forward, but it's in reverse order or backward order.
No, no, that's what now means. Like, let's see. This is this is consider the 20th chromosome. Okay, this is 5 prime to 3 prime. This is 3 prime to 5 prime. Now, PRNP gene. Let's take a random gene now, ABC, whatever it is. So it is located from this to this, and its location is like 5,000 to 8,000. Okay. So this is for the forward strand. You will get the coordinates like a 5,000 to 8,000 base position the gene is located. But the same gene, if it is located on reverse strand, how you will get the number? You are saying like you are getting the number as 8,000 to 5,000, right? This is what you are saying, sir. Correct? This is what you are saying, right?
Yes, ma'am.
And ideally, we want like this. Ideally this is correct because numbering is always from 5 prime to 3 prime. It's not from 3 prime to 5 prime. So ideally, it should be like the number here. It is in a decreasing order. But in a NCBI, you are not getting like that. You are getting like a forward strand only, like 5 to 8. So my question is, in this time, if you are getting a coordinates like this, how can you understand the gene is located on reverse strand? At that time, will you be able to understand then? Will you be able to differentiate if only the coordinates given? Is my question is clear now?
No, ma'am, we won't be able to understand. That is why,
Yeah.
ma'am, I think the arrow is given, I think.
No, but not every time they are not providing the arrow. How they have provided the arrow? So the answer is I'm just opening this PRNT gene in a new tab. For others, the question is clear. I'm asking to everyone.
Can you repeat?
Yes.
What you
Ma'am,
want?
ma'am, the question.
My question is like here as per the for coordinates are given. This is for the forward strand. But for the reverse strand, you will get the same coordinates in ascending order. So my question is, how can you differentiate then the gene is located on forward strand or on a reverse strand? Are you getting my point?
Yes, ma'am.
Hmm. So if I'm checking, I will show you this PRNT gene. Along with the numbering, you will get the complement word here or a C over there. So with the complement keyword or maybe it is providing a C, it means you need to be understand that it is located on complementary strand or on the reverse strand. These coordinates are in a ascending order only. You can see like this is smaller and this is larger, but it is given the complement word. It means it's located on reverse strand. So this is how basically you can understand, you can differentiate. Got it?
Yes, ma'am.
I hope it is clear to everyone. Yes, sir. Is
Ma'am,
it clear now?
ma'am means if the complement word is written, it will be present on the reverse hand.
Right. Complement or sometimes in front of coordinate, here it is given C.
Okay, okay.
Hmm.
Means, ma'am, the reverse arrangement of the coordinates also can be understood that it is on the reverse hand, and if it is
Yeah.
forward, it will be given like descending to ascending.
Yeah, in a forward or reverse, both the coordinates are given in ascending order only, but it just
Okay,
the difference is of the keyword. In forward, you
okay,
will not have any keyword, but in reverse, you are having the complement keyword.
okay,
And
ma'am.
one more
Okay.
thing, I will show you actually during the whenever I will show you the sequence or something. Uh, the sequence you will get as per the forward strand. But still, if you want the reverse strand sequence in the database, whenever you will open the sequence, at the right side, you will have an option that show reverse complement. So when you will click on that option, you will get the actual sequence. Okay? Are you getting my point?
Yes, ma'am.
What about uh-huh.
About the where can we get the sequence?
Yeah, we are coming on that in upcoming part. Let's go step by step. Okay? We are going to everything is for every thing there is a question, practical question. So let's go step by step. For now, the location and the coordinates is clear. Yeah, Annapurna ma'am, is it clear?
Yes, ma'am.
Okay. For the sequence and all, we'll see that in upcoming part. Okay. Is there any doubt to anyone else? I'm sharing the attendance link. Tell me if you have questions. What you can do is, let's solve this question. I will update the PPT in the drive. Go through the PPT and whatever the question we have discussed up to now, just try this for the BRCA one, BRCA one. I have given the name. Kindly mark the attendance. And you can try this question for the BRCA one. Okay? Is it clear?
Yes, ma'am.
Yeah, ma'am.
Okay. Okay. All right then. So that's it for today. The rest of the questions, there are many questions are still left that we'll see by tomorrow.
Okay, thank you, ma'am.
Yeah, you're welcome.
Ma'am, you will send the question or like you will send the PPT? I couldn't hear it.
They will update with the PPT.